students, coming to this video, we will discuss about each part of bacteria. Last class we discussed about what are the parts are present in a bacteria. But this class we will discuss about each part in detail explanation. Okay, right. So the very first one, in the last class we have discussed about the bacteria structure. Within that case, the outermost part of the bacteria is the glycocalyx layer. Glycocalyx layer. See, that glycocalyx layer can be divided into two types. Sometimes the bacteria having S layer, sometimes the bacteria having capsule. Only one is present. Maybe the bacteria having S layer or maybe the bacteria having capsule. It depends on the surrounding conditions of the bacteria. Surrounding conditions of the bacteria. So glycocalyx layer in the normal conditions. Okay, this one is forms S layer. Any bacteria during favorable conditions, the having outermost layer is called as S layer. S layer is easily washable layer. S layer is the easily washable layer. Any bacteria, if we take one bacteria, if we subject that bacteria to the running water, the S layer will be washed out. Because the S layer is very slimy layer. S layer is the slimy layer. That S layer majorly formed during favorable conditions. That S layer, suppose this is the bacteria, that bacteria, around the bacteria, during favorable conditions, the S layer will be formed. During favorable conditions, the S layer will be formed. That S layer majorly having slimy nature. This one formed during favorable conditions. Then what is the use of S layer? Remember that S layer majorly increases the surface area. So the blue line indicates here cell wall. Okay, outside of the cell wall, if S layer is formed, okay, the surface area of bacteria increased. If any cell surface area is increased, then more amount of water molecules are absorbed and more amount of minerals are absorbed. Any bacterial cell we are taken, if surface area is increased, absorption of water molecules is increased and absorption of minerals are increased. Due to the reason is here, see, surface area is increased, right? So that is the major use of S layer. What is the composition of S layer? S layer majorly having high percentage of carbohydrates and less percentage of proteins. S layer made up of with rich percentage of carbohydrates and less percentage of the proteins. This is the slimy layer. Almost any slimy layer, okay, mostly having rich percentage of the carbohydrates. And escape from, see, major use of S layer increases the surface area, supporting for absorption of water and supporting for absorption of minerals. And that S layer also supporting for escape from phagocytosis. The bacteria cells are escape from phagocytosis. Then most of the students have the doubt here, what is phagocytosis? First, suppose in the last class we discussed about WBC. That white blood cells are involved in phagocytosis process. Generally, what is phagocytosis? Right? This is one of the white blood cell. The last class also we have discussed white blood cells are having amoeboid shape, and white blood cells are involved in defense mechanism. Any pathogen or any foreign particle entering into our body, the foreign particles are destroyed. So this is the one of the white blood cell. That white blood cell by the phagocytosis process. Here this is one of the phagocytosis process. During phagocytosis process, one of the bacteria. This is one of the bacteria. So intake of bacteria by the phagocytosis process. Once the bacteria taken into inside the white blood cell, inside the white blood cell, that bacterial cells are destroyed. That bacterial cells are destroyed. So this is the general defense mechanism. This is the general defense mechanism. Simple. One of the white blood cell is there. That white blood cells are having amoeboid shape by the pseudopodias, by the generation of pseudopodias, intake of the bacteria. Once the bacteria taken into inside the white blood cell, that bacteria are destroyed. That bacteria are destroyed. That is the very common procedure. Here I said that slime layer supporting to escape the bacteria cells from phagocytosis process. It's nothing but in presence of S layer, the bacteria cells are escaping from phagocytosis process. So already I said this is on the slimy layer. Presence of that slimy layer, the cells are unable to captured by white blood cells. In presence of slimy layer, that bacteria cells are unable to captured by white blood cells. So simply the bacteria cells are escaping from phagocytosis process. Phagocytosis is nothing but destruction of bacterial cells by the white blood cells. Here, presence of slimy layer, the bacterial cells are escaping from phagocytosis. It's nothing but presence of slimy layer, the bacterial cells are escaping from our immune system. Escaping from our immune system. Once these are escaped from our immune system, which are causes the diseases. The bacterial cells are causes the diseases. If these are killed, we have the immunity. Right? If these are not killed, then bacteria are causes the diseases. Bacteria are causes the diseases. So presence of slimy layer, the bacterial cells are escaped from phagocytosis process. Capsid. So we discussed capsid is a thick coat-like structure. This one majorly formed during unfavorable condition. See the first one, this one formed during favorable condition. This one formed during unfavorable conditions, right? See, especially the major use of capsid protect bacteria cells from desiccated conditions. Protect bacteria cells from desiccated conditions. Desiccated conditions are nothing but dry conditions. Desiccated conditions are nothing but dry conditions. So during unfavorable conditions around the bacteria, this is the bacteria. This is the bacteria. During unfavorable conditions around the bacteria, there is a thick coat formation takes place. There is a thick coat formation takes place. That thick coat is called as capsid. That thick coat is called as capsid. So that capsid is very useful for the protection of bacteria from desiccated conditions. Desiccated is nothing but dry conditions. If temperature is raised in environment also, the water is lost. If water is lost, then whatever that cells are present within that environment, whatever that bacteria cells are present within that environment, then bacteria cells also loses the water. Any cell loses the water, the cell will be dead. The cell will be dead. So protects the bacteria cells from desiccated conditions by the capsid. And here composition of the capsid, high percentage of the proteins are there and less percentage of the carbohydrates are there. High percentage of the proteins are there and less percentage of the carbohydrates are there. And cell survives at a high temperature. I already said, see, especially the thick coat-like structure. Thick coat-like structure, it's the thick coat mainly giving resistance to high temperature. That thick coat or capsid majorly giving resistance to temperature, high temperature, right? That is about the difference between the slime layer and the capsid. Slime layer and the capsid, right? That is about glycocalyx structure. After the glycocalyx layer, the next part is inside of the glycocalyx. The next part is the cell wall. We know cell wall is very important because it majorly gives the mechanical strength to the cell and it majorly gives the structure or specific shape to the cell. So cell wall is the very important structure. Remember, cell wall is the dead part. In any cell, if you have taken any cell, especially in animal cell, cell wall is absent, right? In any cell, like bacteria, like plant cells, like fungi, any cell, the cell wall is dead part. But mostly that is the outermost part. But in bacteria, the very outermost part is the glycocalyx layer, and inside the glycocalyx, cell wall is present. That cell wall majorly made up of with NAM and NAG residues. NAM and NAG residues. See, the NAM and NAG, there is a NAM component is there, NAG component is there. So, NAM indicates N-acetyl murominic acid. NAM indicates N-acetyl murominic acid. NAG indicates N-acetyl glucosamine. In bacteria, the cell walls are made up of with NAM and NAG residues. NAM, here NAM indicates here N-acetyl murominic acid. NAG indicates N-acetyl glucosamine. See, these two components, many number of components, many number of NAM residues, many number of NAG residues are polymerized. Many number of NAM and NAG residues are polymerized. So that polymerization, for suppose NAM, NAG, NAM, NAG, NAM, NAG, right? See, these are forms the one layer, one single unit structure, and another unit structure, and another unit structure, and all these are forms the crosslinks between NAM residues. There is in crosslinks are formed. Finally, all these layers or all these units are combined, produces a cell wall. So many number of NAM and NAG residues, many number of NAM and NAG residues are combined, and finally these are produces the cell wall. And see, this is the NAM and NAG residues are total. The structures also called peptidoglycans. The NAM and NAG residues are the composition of the bacterial cell wall. That is also called peptidoglycans, right? So that is about that cell wall composition, right? See, I already said the mechanical strength provided by the cell wall and gives the shape to the cell. See, most of we are studying MBBS, and we need to know about the antibiotics. What is the relation between antibiotic and bacteria? Just I will explain. Suppose one of the microorganisms enter into inside our body. That time, most of the most of the people are taking antibiotics, right? Once the antibiotic injected into our body, that antibiotics kills the microorganisms, right? Then there is no chance to cause any diseases. Remember, all of you, there is all we know about penicillin. All we know about the penicillin. Penicillin is an antibiotic. First of all, what is antibiotic? Antibiotic. Anti means against. Bio means life. Anti means against. Bio means life. Some of the chemical compounds which are acting against to the life, especially which causes the diseases. Some of the chemical compounds which are acting against to the life, especially that organisms causes the diseases. So penicillin is an antibiotic. Penicillin is an antibiotic. That penicillin, how kills a bacteria? Means my question is here, how penicillin have the capacity to kill a bacteria? Right? Different types of bacteria are there, and different types of antibiotics are there. But my question is, how penicillin have the capacity to kill a bacteria? So further, my answer is here, penicillin, antibiotic, especially it prevents the formation of crosslinks. It prevents the formation of crosslinks. When the bacteria cell wall formation takes place, or during cell wall formation of bacteria, whatever the crosslinks are there, the crosslinks are prevented by the penicillin. Once the crosslinks are prevented, there is no polymerization process. There is no polymerization process. There is no cell wall formation. There is no cell wall. The bacteria will be dead. The bacteria will be dead. So, the simple explanation: penicillin is an antibiotic which mainly act on bacteria cell wall. Which mainly act on bacteria cell wall. Especially prevents the crosslinks formation. Prevents the crosslinks formation. Once the crosslinks formation is prevented, then there is no cell wall formation. There is no cell wall formation. the bacterial cell will be dead. So that's the reason why penicillin is an antibiotic. Penicillin is an antibiotic, right? That is about the chemical composition of the cell wall and functions of the cell wall. Based on the cell wall, the bacteria is also classified into two types are there. There is a gram-positive bacteria, there is a gram-negative bacteria. We'll discuss now. Before discussion about gram-positive and gram-negative bacteria, based on the composition of the cell wall and based on the staining techniques, I'd like to discuss about one important point regarding to the cell walls, especially in prokaryotic cells. We know about all prokaryotic cells are included in Monera, right? That Monera again subclassified into Archaebacteria, Eubacteria, Mycoplasma and Actinomycetes. See, each subclass have the specific character. Here, each one have some very specific characters. Archaebacteria, especially the bacteria which are grown at high temperature. Not only high temperature, the bacteria are grown at unfavorable conditions. Right? Eubacteria, which are grown at normal conditions. Mycoplasma, the bacteria not having cell wall, right, and which are grown at anaerobic conditions. Actinomycetes, these are very different. These are forms the filamentous structures. The bacteria have the capacity to form the filamentous structures, right? These are the total subtypes of Monera classification, right? Within that, Archaebacteria, right? We are discussing about cell wall here. Cell wall. In Archaebacteria, cell wall present. Eubacteria, cell wall present. Mycoplasma, cell wall absent, right? And Actinomycetes, cell wall present. So all bacteria subtypes are having cell walls except Mycoplasma. Mycoplasma not having cell wall, right? And see, Archaebacteria have the capacity to grow at high temperature. Especially in Archaebacteria, a group of bacteria have the capacity to grow at high temperature. These are also called thermophiles. And remaining, most of these bacteria are grown at the normal temperature. Most of these bacteria are grown at the normal temperature. Sir, why we are discussing about these points? Yeah, very important one. Archaebacteria, I said Archaebacteria grown at high temperature. Archaebacteria grown at the high temperature. Remember, the Archaebacteria grown at more than 90 degrees centigrade. More than 90 degrees centigrade. Archaebacteria have the capacity to grow at more than 90 degrees centigrade. Remember, even iron also melted at that temperature. Some of the bacteria are grown at 120 degrees centigrade and some of the bacteria are grown at 150 degrees centigrade. Even iron also melted. So at that temperature also the bacteria are grown. How this is possible? How this is possible to grow bacteria at very high temperature? How the thermophiles have the capacity to grow at the high temperature? Because the very first reason is composition of cell wall. Composition of cell wall. The thermo indicates here temperature, right? Philes. The bacteria which are grown at high temperature, these are called as thermophiles. So thermophiles which are grown at high temperature, because one of the reason is composition of cell wall. See, the compound name is pseudomurin. The compound name is the pseudomurin. Pseudomurin is the cell wall composition and branched chain lipids. Branched chain lipids. See, especially in cell wall, pseudomurin is present. Because of the pseudomurin, right, the bacteria are grown at the high temperature. And along with that pseudomurin, the branched chain lipids also present. Long chain, very long chain, right? Branched chain lipids also present. That branched chain lipids, especially these are seen in plasma membrane. That branched chain lipids which are seen in plasma membrane. So in cell wall, pseudomurin is present. Because of pseudomurin, because of branched chain lipids, the thermophiles which are grown at the high temperature. And composition of eubacteria we already discussed. NAM and NAD, that is nothing but peptidoglycans. So eubacteria, the composition of the cell wall is the peptidoglycans. Mycoplasma, there is no composition here because here cell wall is absent. In Mycoplasma bacteria, No composition of the cell wall because cell walls are absent. Actinomycetes. Within actinomycetes, the composition of cell wall is mycolic acids. Mycolic acids. So here within Moneria, different types of bacteria are there. According to the subtypes, the composition of cell walls also altered. The composition of cell walls also altered. In all these points, the very important point is archaebacteria. Within that archaebacteria, a group of bacteria are there which are grown at high temperature. These are called as thermophiles. Why these have the capacity to grow at high temperature? Because the reason is that cell wall composition, and cell wall composition means cell wall contains the pseudomurine, and especially within that plasma membrane there is a branched chain lipids are there. Because of pseudomurine and because of the branched chain lipids, the bacteria are grown at the high temperature. Right? So that is about different types of the subclasses of the Monera, and compositions of different types of bacteria.